My experiment was specifically show how effective different soaps are. I am conducting this experiment because of an interest in different types of soaps and how effective they are on common bacteria. This topic is academically important because it gives insight into what concentration of certain active ingredients, in this case triglycerin, is the most effective at inhibiting the growth of bacteria. It is also important because it can give people more knowledge on the effectiveness of the soaps that they're using on an everyday basis. My hypothesis is that soaps with a greater concentration of the active ingredient triglycerin will be more effective in inhibiting the growth of the bacteria E. coli and S. epi. The materials and equipment used in this experiment include the selected bacteria and soaps, petri dishes, agar solution, sterile swabs, an incubator, sterile water, filter paper, alcohol, a ruler, and Excel. The bacteria chosen for this experiment were E. coli and S. epi because they are relatively common bacteria and have different cellular structure. E. coli is a gram-negative bacterium, meaning it has a cell wall made up of several layers of peptidoglycan and is usually more resistant to antibacterials. S. epi is a gram-positive bacterium, so it has a cell wall made up of a single layer of peptidoglycan and is more susceptible to antibacterials. The soaps were chosen based off of their concentration of triglycerin, with it ranging from none to a high concentration. To begin this experiment, you will first need to obtain all of the listed materials and equipment. You will then need to transfer the bacteria to a petri dish using a sterile swab. Next, you cover the filter paper in the selected antibacterial and place the treated filter paper onto the bacterial site. Then seal the petri dish and place it into the preheated incubator. You will then need to leave the petri dishes in the incubator for 2 to 3 days to allow them to fully grow. Once you have done this, remove the petri dishes and record your results in Excel. The results from the experiment showed that a greater concentration of the active ingredient triglycerin was more effective in inhibiting the growth of E. coli and S. epi. The experiment was run 4 times to ensure accuracy, and then a t-test was conducted to test the null hypothesis, which was that there is no difference between the growth of bacteria that are or aren't treated with soaps containing triglycerin. After setting the level of significance at p is less than 0.05, the null hypothesis was able to be rejected when the treated bacteria, which had a mean of 1.21 centimeters, showed a statistical difference from the control group, which had a mean of 2.40 centimeters. This graph shows the raw data collected, with every 4 runs on the x-axis being a different type of soap and the y-axis being the diameter of the zone of no growth in centimeters. The results for E. coli are shown in blue, while S. epi is shown in red. After analyzing the data, it was found that some soaps had no effect on the bacteria, while others were able to completely inhibit their growth. These results can be contributed to the concentration of triglycerin in the different soaps. Also, the different cell structures could be seen in the gram-negative E. coli's ability to be more resistant to triglycerin than that of S. epi. This picture gives an example of how the petri dishes were labeled before they were placed into the incubator. This picture shows the effects of allowing the bacteria to grow for a day, with the 4 circles around the filter paper being the zones of no growth. This picture shows how the bacteria grows when there is no antibacterial present to inhibit its growth. I would like to thank everyone who supported me during my project and also my supervising and assisting teachers, Dr. Zhu and Mrs. Hamilton.